Hey biology students! In this lecture video, we're going to discuss the replication of RNA viruses. So pictured here on the screen, we see the influenza virus. And the influenza virus is an example of an RNA virus. You'll also notice it's an enveloped virus. So we see it has a genetic core of RNA, and then it has a capsid. Now it has a spherical capsid, and then it has a lipid envelope and several different spike proteins that are sticking out from the surface of the influenza virus. And we see on the left what that looks like under the electron microscope. So in the last video, we learned about the 6 stages of the viral lytic cycle. In this diagram, we're seeing a very general picture of those stages, of course without the labels. So let's put the labels in. So remember, this is 6 stages where the outcome will be cell death and release of many viral progeny from the host cell. So the 6 stages are, as a review, attachment. This involves a specificity between the viral ligand, which is usually some sort of capsid protein or spike protein that attaches to the host cell. This then triggers a chain reaction called penetration, and we learned about 2 mechanisms of penetration. We have receptor-mediated endocytosis, which is a process that occurs in both the naked viruses as well as the enveloped viruses. And then we have a process called fusion, which is found only in enveloped viruses. And we watched animations of those 2 different mechanisms for penetration in the last video. The next stage is once the virus gets inside, okay, so let's say it's engulfed in endocytosis, and remember that involves the formation of an endosome, then what happens is then the viral RNA is released into the cytoplasm of the cell. And that involves uncoating. So what happens is the capsid is released or dissolved, okay, uncoating to release the viral. Now we'll just keep it general here. We'll just say viral nucleic acid from the capsid. So that's the uncoating stage. Okay, the 4th stage we call the synthesis stage. So the synthesis stage, in this picture, we're seeing lots of copies of viral RNA or viral DNA. The nucleic acid of the virus is being copied. And then viral proteins are also being made. So we're seeing new viral proteins, and we're seeing new viral nucleic acid being copied. So it's putting all the parts of the virus into the cytoplasm, and then, like going to IKEA and building a dresser, assembly is required. So step 5 would be the assembly stage. So this is putting all the pieces together of the virus, package that viral genome into the capsid, put the capsid or put the envelope spikes into the membrane of the cell, and then we have the 6th stage, which is the release stage. Okay, and so this involves the new viral progeny being released from the host cell, and of course in the lytic cycle, this will always end in death of the host cell. That's of course what the word lytic means, which means to break. And what's going to break? Well, in this case, the host cell. So as a result of the lytic cycle, the host cell dies. New virus is released, and then that virus goes on to infect a new cell, and the cycle repeats. So in the last video, we focused on how this works in a double-stranded DNA virus. And remember, the double-stranded DNA virus uses two of our cell enzymes, the DNA polymerase to copy its DNA genome and make more DNA, as well as RNA polymerase to copy the viral DNA into messenger RNA that can then be translated by the host cell's ribosomes and make viral proteins. Okay, so now I would like to tell you about how RNA viruses replicate during the lytic cycle. So an RNA virus is going to follow the exact same 6 stages of the lytic cycle, but where the variation is going to be is at the synthesis stage. So the synthesis stage is going to vary for DNA viruses versus RNA viruses. Now, the first thing I would like you to know is that there are 3 different types of RNA viruses. So we're going to now go through each type, and we'll talk about how the synthesis stage works for that particular RNA virus. Now, it's going to get a little complicated here. So remember, you can always pause the video, and you can always rewind and watch it again. This is a complicated step, okay? Just hang in there. So 3 different types of RNA viruses. So let's start with the first type. So the first type has a positive-sense RNA. Okay, so number 1. In other words, what this means is that it's a single-stranded RNA virus. So what that means is that if it's a positive-sense RNA virus, what that means is that the way the viral RNA is structured, it can be directly translated into protein. In other words, this single-stranded RNA acts as viral mRNA. It's read directly as viral RNA, okay? So that means that when the virus is uncoded into the cytoplasm, so in stage 3, it's uncoded to release the viral RNA, that viral RNA does not need to enter the nucleus, and what happens is that RNA is directly translated by the ribosomes of the cell. So here we have pictured the ribosome, the cell's ribosomes, and we're going to show that the viral positive-sense RNA is being used as mRNA. So it's essentially mRNA. That's what it means to put the little positive next to this RNA. It doesn't mean that it has a positive charge, okay? A lot of times I see students misinterpret what we mean by that. It doesn't mean that it has a positive charge. It just means that it can be directly translated into protein. It's positive sense. In other words, it makes sense to the cell. The cell reads this messenger or viral RNA as a viral messenger RNA. It's already written as sense for the cell. So it's directly translated into viral proteins. So translation is immediately initiated, and as a result, we get production of viral proteins. And so that could be capsid, that could be viral spikes, okay? So remember, that is one of the goals in the synthesis stage, the goal being to create many copies of viral proteins that can be assembled into new virus. But what's the second goal? That's right, the second goal is to actually copy the viral genome. In this case, the viral genome is positive-sense single-stranded RNA. So how's that going to work? Well, that means that we need to take this single-stranded RNA and we need to make many copies of that can be packaged into new virus. So we need to take RNA and we need to copy it to make more RNA. Does the cell, our cells, do we have an enzyme that can copy RNA to make more RNA? That's right, we don't. We have enzymes that copy DNA into DNA called DNA polymerase. We have enzymes that copy DNA into RNA. We call those RNA polymerases. We do not have the ability to copy RNA into RNA. So that means we need an enzyme to do this. Well, where does this enzyme come from? Well, it actually is one of the proteins that's translated over here. So let's add one other thing here. We're going to make it an R, okay? And that's for the name of this particular enzyme. So this is, remember a lot of times we show enzymes sort of looking like Pac-Man. So how about that? So this is the enzyme. This is a viral enzyme in purple. The name of this viral enzyme is RNA replicase. So let's write it over here. So it comes from, so in other words, the virus has to translate the proteins over here. An enzyme is a type of protein, so it gets this protein from over here, and it needs that in the replication of its genome step. Okay, so this is called RNA replicase, and it is made by copying the viral RNA and making viral proteins. So in other words, it's viral origin. It's not a normal enzyme that our cells have. Okay, so it's called RNA replicase, although it does go by another name. And just so you're aware, there is another name. So we're going to say it's also known as RNA. This is a longer name, okay? It also is known as RNA-dependent RNA polymerase. Which is abbreviated RDRP. RNA-dependent RNA polymerase. That's the same thing, just different name. In other words, it's telling you it is an RNA polymerase, but it's RNA dependent. In other words, it uses RNA as its template to copy and to make more RNA. And that's different from the RNA polymerase in the cell. So the RNA polymerase in our cells is a DNA-dependent RNA polymerase, meaning that the RNA polymerase that we have copies DNA into mRNA. Yeah, I know it's confusing, which is why I prefer to go by the name RNA replicase, which means to replicate RNA, and I find that's a little bit easier for students to follow. But just in case you stumble across this other term, you now know what that means. So it is replicating the RNA of the virus. Now the way it's going to do that is it's actually going to use that single-stranded RNA sense copy, but it's going to produce a reverse complement of it that we call the antisense. And we put a little negative there to mean that's the antisense copy. So it's going to copy the sense into the antisense copy, and then it's going to use the antisense copy as a template to make more sense copy. So you have to go from sense to antisense to make more sense. Yeah, I told you it was complicated. Pause it, rewind. Look at this. Bullet point, translation. Okay, we drew that here. So we drew that the translation step produces viral proteins, and one product happens to be the viral RNA replicase, which is here. And then that viral RNA replicase acts as the enzyme to make copies of RNA. In order to make more of the single-stranded RNA, you need RNA replicase. And it's synthesized by the translation process. Okay. Now let's look at the second type of RNA virus that I would like you to know. The second type is called the antisense RNA. So the antisense RNA, it has a single-stranded RNA genome. However, it is opposite in orientation to what the cell would use in order to make viral proteins. So it has to copy its antisense. So we put this little negative here, which means antisense. It doesn't mean, again, that it has a negative charge. Okay, so that's not what it means. It just means that it's not going to be directly translated. So we usually put a little negative symbol next to it there to mean that it is the antisense copy. Okay, or we write it this way. So single-stranded RNA that is antisense. So that means that after the uncoding stage, the antisense RNA gets dumped into the cytoplasm of the cell, and the cell has no way of dealing with it. It doesn't know what to do with this antisense RNA. So that means that this type of RNA virus, so we're saying this is number 2, this second type of RNA virus has to have a way to copy its antisense RNA genome into sense. Okay, so how's it going to do that? See where I'm going with this? So we're going to copy the antisense RNA genome into a sense genome. Remember, we use the plus sign to mean that it's the sense genome. So we need to copy it into the sense genome. So do we know of an enzyme that copies RNA into RNA? That's right, RNA replicase. So that means RNA replicase has to be packaged with this particular type of RNA virus. So rather than, back up a minute, back to the sense RNA virus, okay, this type of virus here, okay, where what happens is, remember, this means that it's being read directly as viral mRNA. It makes sense to the cell. The cell says, oh yeah, that makes sense. And directly translates the viral mRNA into viral proteins. And one of those viral proteins happens to be this viral enzyme. And then the second step would be this viral enzyme would come over here and would start copying this viral genome. So the difference is that the antisense RNA viruses, these types of viruses have to come packaged with RNA replicase. Otherwise, they would have no way of replicating because, again, if it's antisense, the cell has no way of dealing with that particular type of viral genome. Okay, So we rely on this RNA replicase, very important enzyme, very important viral enzyme that copies RNA into RNA. So it'll go RNA into, or antisense RNA into positivesense RNA, which is basically viral mRNA. And then that will allow for translation to occur. So then it will go to the ribosomes, and we would get translation. And we would get viral proteins, so capsids, spikes, and of course, the RNA replicase enzyme. Then that RNA replicase enzyme would also be important in making more copies of the antisense genome. Remember, what it's going to do is it's going to copy that. Oh, that was a really bad one. So remember, what it's going to do is it's going to copy the antisense into sense, and then it'll copy the sense into more antisense. It'll use that as a template. Okay, so the difference here is that that viral RNA replicase has to be packaged in the virion. So what is that going to look like? That means that in the assembly stage, we are going to have single-stranded antisense RNA as the viral genetic core, and that's going to be surrounded by a viral capsid and also inside of the capsid we would find the enzyme, the RNA replicase enzyme. Very important enzyme that's facilitating this step, and we might as well draw it in here. It's doing this as well. So it copies antisense to sense and then sense to antisense to make a whole bunch of antisense genome and package that into the capsid. And now if this is an enveloped virus, then it may have an additional envelope around it with then the viral spikes that would be added once the budding process occurs. And this is exactly what influenza is. So influenza is an antisense RNA virus. Okay. Third type of RNA virus, retroviruses. So retroviruses are RNA viruses, but they copy their viral RNA into viral DNA. And in order to do that, they come prepackaged with the viral enzyme reverse transcriptase. So the example I wanted to give you was HIV. So HIV is an example of a retrovirus. So, for example, okay, so what happens is the HIV is actually a positive single-stranded RNA virus But that positive single-stranded RNA virus, the first step in the life cycle of HIV is to then copy its viral RNA into DNA. It's viral double-stranded DNA. So it's copying from RNA into DNA. And that's the reverse of transcription, which is why the enzyme that facilitates this process is called reverse transcriptase. So it also has to be prepackaged with the HIV virion. We'll call this enzyme RT for reverse transcriptase. So the reverse transcriptase enzyme is utilized by the virus to copy its genome from RNA into double-stranded DNA. And it does this because many retroviruses, like HIV, actually integrate their DNA into the cell's chromosome and goes through a latency period. And in order to go into a latency period, it needs to copy its DNA or copy its RNA into DNA in order to do that step. Now, let's think about comparing these RNA viruses with double-stranded DNA from the last lecture. Go through each one of these questions and think about what the answers would be. So question 1, does the virus use the cell's DNA polymerase? Well, let's look. Double-stranded DNA. So a double-stranded DNA virus has to copy its DNA into DNA, more DNA. So yes, it's going to use the cell's DNA polymerase enzyme to make that possible. Will the RNA viruses use DNA polymerase? No. Okay, so they have no need for the DNA polymerase. Number 2, does the virus use the cell's RNA polymerase? In other words, is it going to take the DNA and copy it into RNA? And that would be a yes for the double-stranded DNA viruses. They are going to use RNA polymerase from the cell to facilitate that. What about the RNA viruses? Do they use RNA polymerase? Not typically. Okay, not to copy their genome. In the case of those retroviruses, though, that become double-stranded DNA, then yes, they would use the RNA polymerase from the cell. Number 3, does the virus use the cell's tRNAs and ribosomes to make viral proteins? So do we make viral mRNA that then is added to the ribosomes, and then we make viral proteins? So yes, everybody uses that. Everybody is going to rely on the cell's translation machinery in order to make more viral proteins. Number 4, does the virus make the viral RNA replicase? That is the enzyme that copies RNA into RNA. The double-stranded DNA virus does not have use for that, so no. The single-stranded RNA virus does copy RNA into RNA, the function of RNA replicase, so yes. And the antisense RNA virus also copies RNA into RNA to make more copies of its genome, so yes. The difference is that with the antisense RNA, it comes packaged with RNA replicase. Okay, so I know that is a lot of information, so Now go ahead and answer your study questions and rewind if you need to review the ways that RNA viruses replicate.